AS Biology. Welcome. Are you excited for a fun learning session? Make sure you have your book and a pen before we start. Today you are going to learn about DNA replication. DNA replication. DNA strands are antiparallel. This means that the DNA strands run in opposite directions so that the bases meet. When cells divide, two new daughter cells are formed, and so DNA must replicate to ensure that each daughter cell has a copy of the DNA in its nucleus. Semi-conservative replication means that each new strand of DNA contains one strand from the original strand of DNA. This helps to ensure that the DNA in the new cells is accurate. This provides genetic continuity for the organism. The new strand is built from the template of the old strand. This is because of complementary base pairing. In semi-conservative replication, each new strand containing one strand from the original. Watson and Crick came up with the theory of semi-conservative replication, but it remained a theory until an experiment was done by Meselson, and Stahl did an experiment in 1958 to test the theory. The Meselson and Stahl experiment involved: Step 1, growing two samples of bacteria in nutrients. One sample contained a heavy isotope of nitrogen N15 and the other contained a light isotope of nitrogen N14. As the bacteria replicated, they used the nitrogen from their sample to make nucleotides for their DNA. Gradually, the two samples incorporated the nitrogen into their DNA. Step 2, a sample of the bacteria from each sample was taken and the DNA extracted. This extracted DNA was spun in a centrifuge. The DNA with the heavy nitrogen settled lower than the DNA with the lighter DNA. Step 3, the DNA from the heavy nitrogen was taken and put into the light nitrogen. Step 4, the bacteria was left to complete one growth cycle, so that they would take up the light nitrogen into their DNA. Step 5, then a sample of DNA was taken from these bacteria and spun in a centrifuge. Step 6, the DNA settled halfway between where the heavy DNA had settled and where the light DNA had settled. What did this mean? If when the bacteria had divided, one bacteria had taken all the original, heavy, DNA, that DNA would have sunk to the bottom, and the other bacteria would have DNA entirely from light DNA, and this would have settled at the top, forming two bands. As all the DNA settled in the middle, it showed that the DNA contained both light and heavy nitrogen. If DNA replication was conservative, then there would have been two bands showing heavy, bottom, and light, top. However, because DNA replication is semi-conservative, there is only one band of DNA in the middle. The first stage of DNA replication, the helix unwinds and the hydrogen bonds between the bases break to separate the DNA into two separate strands. Each separated strand now acts as a template for a new strand. Free-floating DNA nucleotide bases will be attracted to their complementary base pair and the new strands will form. A condensation reaction will bond the nucleotide bases together to form the new backbone. This process uses an enzyme called DNA polymerase. Hydrogen bonds will form between the complementary base pairs. Each new strand of DNA has a strand of DNA from the original and a newly formed strand. DNA strands run antiparallel and the ends are named as each end has a slightly different structure. One is 3' and the other is 5'. Due to the slightly different structure of the 3' and 5' ends means that the DNA polymerase can only move from the 5' end towards the 3' end. This means that the DNA polymerase works in different directions. When the strands are split, one DNA polymerase will work from the 5' to the 3' on both strands.